break. You didn't have to worry about molecular biology at all. Probably makes this morning a little bit rough, but that's all right, it'll get easier. Has everyone purged all the molecular biology information from their brains at this point? Took an exam and just complete data dump. So, last time I saw you guys, we had an exam. That exam is not done. I'm not done grading them yet. Hopefully we'll get them done by Tuesday, tomorrow. So you'll have that score by the end of the week. Just like before, I'll be doing an update on everything up until that point. I know there are still some grades that I haven't updated from the last round. I'm still getting through all of those, so have some patience with me. I'm going to try to get everything corrected by the end of the week so that we're up to date with everything. And I'll let you know once everything's been updated as far as I'm aware of, and then you can take a look and make sure I've gotten everything correct. And we'll go through that same process of point adjustments, making sure we've gotten everything recorded correctly, we haven't missed anything, etc. Okay? So once we get the exams graded, hopefully you'll get those back in recitation this week. And then I'll let you know at the end of the week sort of what time frame you have for point adjustments. If there are any questions as we grade them that I come across that I want to make point adjustments for everyone on, etc., just like we've done before. Okay? One quick announcement. I made an announcement right before spring break about a research opportunity in my lab. I just sent out those applications yesterday. If you didn't get one and you wanted one, or you emailed me and I missed sending it out to you, please let me know. I'll be looking for those applications to come back in at the end of the week, and you'll hear from me from there. Okay? Exam theory we already talked about. Any questions about the last exam? Grade updates before we continue? Completely forgotten everything about it. So no questions. All right, good. Then let me remind you what we were talking about right before the last exam and before spring break. We had just finished talking about mitosis and meiosis. The last exam, you had to be able to compare and contrast those, recognize any stage of either cell division process. We're going to go from starting here. So today we're going to review just a few topics we covered rather quickly at the end before the last exam. And then we're going to start to get into Mendelian genetics, just very, very briefly, as a preview for what will be coming up when you take genetics. Next year, I think you take genetics. You'll see a lot more of Punnett squares and alleles and things like that. So we finished up talking about mitosis and meiosis. And in particular, we were looking at what happens in anaphase 1. And we looked at the ways that pairs of homologous chromosomes arrange themselves. And we talked about how there's independent assortment. So each pair of chromosomes lines up independently of each other pair, so that there's no association between chromosomes and how they end up in the gamete. Right? We call that independent assortment. And that has ramifications for the different kinds of gametes that can be produced from a cell in that process of meiosis. We'll come back to that in just a minute. I also just want to make sure that we're all on the same page as far as mistakes in the process of meiosis. We talked about non-disjunction and what happens when either homologous chromosomes or sister chromatids, so anaphase 1 versus anaphase 2. And if they fail to separate properly, what happens? All right, so we get everyone's brain going. Yeah, click your questions. All right, so non-disjunction. When things fail normally during anaphase, what happens?
Wait, oh no, this was the icon courses.
We talked about this briefly. Think about what happens when things fail to separate. What does that mean for how the chromosomes end up in the gamete?
Oh, I didn't see German.
Everyone get a chance to answer that one? This one you've already seen at least once. Trying to jog your memory. You want me to get their answers in? Let's take a look. So, in non-disjunction, it can result in didn't answer. All right. You guys are awake. Excellent. All right, so it is E. Right? It can result in gametes that have too few chromosomes, too many chromosomes. It can happen in either female or male germline. All right, so if this is our normal process of meiosis, what's the diploid chromosome number in this cell? 4. Thank you. Right? And through the normal process of meiosis, we end up with 4 gametes, which each have a haploid number of 2. So starting with that same diploid cell of 4 chromosomes, you guys should be able to draw. So draw now.
What?
All of the gametes produced as a result of a non-disjunction event during anaphase 1. So when you get one pair of chromosomes that don't separate, and all of the gametes produced from non-disjunction at anaphase 2, when you get a pair of sister chromatids that don't separate. What do those gametes look like in terms of chromosome number? Too much this early in the morning? All right, anaphase 1, homologous chromosomes. Anaphase 2, sister chromatids. Think about how many chromosomes are in each of those stages. And if they don't separate, what does that mean for the gametes that are formed? That, but it's likely that that cell won't be viable. At least in animals. Plants. All right, so we're just talking about non-disjunction events of one chromosome, or one pair of chromosomes. Draw it out. Where do they end up? You have figured out? You're looking stumped. So draw out anaphase 1 and anaphase 2. Should be able to do that for the last exam. And how the chromosomes, how many chromosomes there are, what replication state they're in, and how they're lined up. And then move one pair of those so that they don't separate into a gamete. See what it looks like.
No.
Certainly possible. Sorry? No, treat anaphase, non-disjunction anaphase 1 and anaphase 2 as separate events. Yeah, not at anaphase 1 and then at anaphase 2. Sorry, let me clarify. Two separate cases.
Okay.
Alright, I'll give you a few more seconds. I'm going to ask you a question about it next, so make sure you have your drawing. Alright. Everyone have their gametes figured out? Alright, so you see this set of gametes, where did non-disjunction occur? Anaphase 1, anaphase 2, neither of those or both of those. What produces this outcome?
Oh, that means you have extra.
Got it? Excellent. All right, everyone get their answers in? One more second.
I knew the album.
All right, get your answers in. Please take a look. Okay. So the non-disjunction occurs at, right, the class thinks that it occurs at anaphase 2. That is correct. So let's go through and take a look at the difference between anaphase 1 and anaphase 2 and the results of non-disjunction. All right, so here's our normal meiotic division. We get separation, division 1, separation of our homologous chromosomes, 2 of our sister chromatids to produce 4 haploid gametes. If in meiosis 1 and anaphase 1, we get a pair of homologous chromosomes that don't segregate, then we end up with too many chromosomes in one cell, daughter cell, and not enough chromosomes in the other. So we end up with a set of gametes that are all off in some way. So we get two that have an extra chromosome relative to haploid number, and two that have one fewer chromosome than haploid number. So all the gametes have mistakes in them. If we look at non-disjunction in anaphase 2, so we don't get the separation of a sister chromatid. We still have one daughter cell from that first division that separates correctly. So we have the possibility of getting two normal gametes, and then the gametes from this cell, where the chromatids didn't separate, we get one with too many and one with not enough. So what is the more serious non-disjunction event? Anaphase 1, it affects all the gametes. The more mistakes that there are, the less likely those gametes are going to be viable. Right, someone asked, it can happen in both anaphase 1 and anaphase 2. And you might possibly end up with a gamete without any chromosomes. That's not really a viable gamete. So there are checkpoints in place to make sure things have separated correctly, but there are mistakes made. Some of the most common ones. So Down syndrome is trisomy chromosome number 21, which I don't have on here. Some other ones involving the sex chromosomes. So we have Klinefelter syndrome, where an individual ends up with an extra X chromosome. So they have one extra chromosome, 47 chromosomes instead of 46. Right, so this individual would be considered a male, but because of that extra chromosome, have issues with sterility and some of the other sort of sex phenotypes. Here's one that's an example of missing a chromosome. So this is it's considered a female, but she's missing one copy of the X chromosome, so she has 45 chromosomes instead of 46. And there are also ramifications and phenotypic results from missing that X chromosome. All right, so it does happen occasionally, and it results in these syndromes that have often developmental and sex-related phenotypes, right? So they have important effects on the development of an individual. Okay, so that's what happens when things go wrong. Let's go back to thinking about what happens when everything works properly. So we had this question, I think, in class the week before spring break. Right? Another jog your memory. We looked at, related to that independent assortment of chromosomes, how many different types of gamete can be made from this particular cell. Diploid number of 4 chromosomes. And by types of gametes, I mean how many different ways can those chromosomes assort into gametes? How many different ways through the process of meiosis combinations can you put these chromosomes together? All right, count them up. We're going to ramp up to increasing numbers of chromosomes. Let's see if you can figure out the pattern. All right, I'll give you one more second. All right, all the different possible combinations of 2 chromosomes of our homologous set. Let's take a look. Okay. So 4 is the correct answer. 2 sets of homologous chromosomes can assort independently to result in 4 different possible gametes. We look at how that happens, just to give you a visual. So possibility 1, they line up like this. They sort into sort of the blue side and the red side. And you end up with gametes that look like that. Possibility 2, they sort opposites to that. You end up with a blue and a red in each gamete. 4 possible ways that chromosomes can end up in gametes. We're talking about 2 pairs of homologous chromosomes, a diploid number of 4. So what is the relationship between the number of chromosomes and the number of gametes that can be made? All right? We've looked at a simple case. What about if we looked at the set of chromosomes in the human genome? How many different gametes can we make from 23 pairs of chromosomes? Right? We could do what we did before and count them up. Right? So here's example number 1. Should we go through all the examples? That would take the rest of the semester, I think, right? So let's start with a smaller number. We'll work our way up. So we already looked at this example. So for the case of counting our types of gametes, we're going to look at chromosome pairs, and we're going to call them left and right for each version of that chromosome, of that homologous pair. So we already know that there are 4 ways that we can put these 2 pairs of chromosomes together into gametes. We can end up with a left-left, a right-left, a right-right, and a left-right. So 4 possible ways we can make gametes from these 2 pairs of chromosomes. What if we add another pair of chromosomes? We have a diploid number now of 6. How many gametes can we make?
8.
Why?
2 possible combinations each.
2 possible combinations of each. So we had 4 possible combinations that we can add the new left chromosome to, and 4 possible combinations that we can add the right chromosome to, for a total of 8 gametes. What about if we added another one? How many gametes? Using the same logic? Heard it over here. 16. 8 possible combinations plus left. 8 possible combinations plus right. 16 different gametes. We added another one. You guys getting the pattern? All right, so how many gametes for 5 sets of chromosomes? 32. 16 plus left, 16 plus right. So what's the formula? 2 to the n, where n equals the number of chromosome pairs, or another way to say that, number of sets of homologous chromosomes. Okay, so 2 to the n, 32 for 5 sets. All right, so go back to the scenario for humans. We have 23 pairs of chromosomes. How many gametes can an individual make with that set of chromosomes? Who's got a calculator? A lot. Roughly 8 million. Right? So 2 to the 23. 8 million different gametes. Because we're diploid individuals, we undergo sexual reproduction to produce progeny. That means we have 8 million possibilities for our gametes, times 8 million possibilities for the gametes that are joined in sexual reproduction for a whole lot of combinations. So sexual reproduction produces vast amounts of genetic diversity by mixing up sets of chromosomes. All right, and then don't forget, we're only talking about counting up possibilities when there is no recombination. Right? This only works if there is a recombination. If we start accounting for recombination, right, that's swapping of bits of non-homologous sister chromatids that happens in the first meiosis, in prophase, then you end up with astronomical amounts of ways that you can shuffle genetic information. All right? So the process of meiosis, like going from diploid to haploid cell, in conjunction with that recombination of genetic material, we get vast amounts of shuffling of that genetic diversity that's then passed on to the next generation. All right, so that leads us up to thinking about genetics. So that's the process that produces these gametes, these possible sets of genes that are passed on. So genetics is thinking about how those gametes are recombined into the next set of the next generation. All right, so hopefully you're all familiar with Mendelian genetics to some degree. You're going to get a lot more of it when you actually take genetics. But we're going to, over the rest of today and a little bit on Wednesday, just introduce you to some of the basic concepts. All right, and we are talking about diploid organisms. Organisms that undergo sexual reproduction, that have 2 copies of each chromosome. All of the metazoans. The diversity of animals, for the most part. There's always exceptions, of course, because it's biology. All right, all of these organisms are diploid, which you should recognize at this point means that they have 2 copies of the genetic material. Which means they have 2 copies of every chromosome, which means that they have 2 copies of every gene. And of those copies, one comes from the mother and one comes from the father. We're going to start looking at how those copies come together. If we have 2 copies of every gene, what that means for the trait that that gene codes for. What's the interaction between those 2 copies? All right, so let's bring it back to our current understanding of what a gene is. So our current understanding is a molecular definition. What does our molecular definition of a gene consist of? If I asked you to draw a gene, what would you put up there? You have a promoter, you have a transcription unit, regulatory regions, initiation, termination. All right, it's a very, I'm going to say concrete definition of a thing in our DNA. That's what our drawing of a gene looks like. We're going to start talking about a Mendelian definition of a gene. All right? The Mendelian definition of a gene is a unit of inheritance, something that can get passed on to the next generation. So they're related, but we've added another layer on it. That's something that is passed on. And we're specifically going to think about now for diploid organisms this idea that individuals have 2 copies of every gene. So if we have 2 copies, what do we call each copy? An allele. So how would you define an allele?
One copy of a gene.
Different variations of the same gene. Right? Those 2 copies don't have to be identical, and as a matter of fact, generally they aren't. So an allele is an alternative form of the same gene. And in each diploid cell, we have 2 alleles. We have 2 copies of everything. So we want to think about how we get those 2 alleles and the interactions between those 2 alleles, resulting in phenotypes, the ultimate characteristics of the organism. All right, so here's our molecular definition of an allele now. Some version of a gene. So this is a version of a gene. It's one allele. Let's think about how you can create different alleles of our molecular concept of a gene. All right, so in this version, I've shown you 2 genes. That blue line represents a place where the DNA is different. It can be as small as a single base pair difference. So look at where that difference is in relation to the concept of the gene. Are these 2 genes different alleles?
Well, sometimes, yeah.
You have black hair, you have black hair. You guys might have different.
I'm not TV.
All right, think about our molecular definition. That blue line, single base pair difference.
What happens is, if we always change the mRNA,
Is that a different allele?
oh. No? No, I said. So it's deep.
Yeah, good question.
Because that could just be a random normal vector sequence.
The only information you have is the DNA sequence. And there's a 1 base pair difference.
So deep.
That's the question.
Well, what's the question? Yeah,
So if you change the DNA but you don't change the product, is that still the same allele? What do you think?
It's splitting into the legs there. Okay, right.
Because it's hard to believe that every theoretical wise.
Oh, okay. I didn't see.
All right, everyone get their answer in? Let's take a look and see what the class thinks. So the question really is, I'm not giving you any information about how that nucleotide is related to the protein or the phenotype. I'm just asking, is a 1 base pair difference, regardless of any other of that information, is that still considered an allele? All right, so let's see. Not that. Oops. Crap. Wrong button. I'm not sure I can get back to the results now. All right. So, who voted for yes? attended yes? Who voted for no? Who attended no's? Who voted for depends on whether it changes the mRNA? Who voted for depends on whether it changes the protein? Who voted for E? You have no idea what I'm talking about.
Oh, I didn't like that.
Okay, so the correct answer is, it doesn't matter whether it changes the protein. It's still a different variant of the gene. It's still a different allele. The DNA is different. Yeah? Yes? You get that? So the answer is A. Yes, these are different alleles.
You think about it.
I have another version of it now. All right, so this particular change is in the transcription unit, and it doesn't matter whether or not it changes the product. It's definitely going to change the mRNA, right? Because you've changed the nucleotide sequence, so the mRNA is going to be different. We have no idea whether the protein produced is going to be different or whether that different protein is going to affect the phenotype. But it still counts as a different allele because it's a different DNA sequence. All right, what about this particular change? What if we move that one nucleotide base pair change to the promoter?
Does it screw up the promoter?
Don't know. Is it a different allele? Does it matter?
No, I think
All right, remember this blue line can be a single base pair change. It can be a change of 10 base pairs, 100 base pairs. It can be a deletion of base pairs. Doesn't matter what the difference is. There's a difference there.
It's still good. Like, so it's like, I don't know.
Think about what our definition of a gene is. Think about our definition of the allele. Does
It's
this
just like there's a
change
gene in the transcription.
make these two different alleles or not?
So is it A? Thinking it's either A or B. Yeah.
Get your answers in.
The grip is so like big.
All right, let's take a look.
Oh dear. Just goes.
Tom will get it right. There we go. All right, so split decision between yes, they are different alleles, and no, they are not different alleles. Correct answer?
Wow.
Charles is unhappy with that. All right, they are still considered different alleles. Our definition of a gene, the molecular definition of the gene, is everything between those red lines. There's a difference between them. A single base pair difference is in the promoter. It may affect promoter binding of the transcription factors, or it may not, but it's still a different variant of the gene. There's a difference in the DNA that codes for it. One more? You guys ought to be able to figure this one out now, right? What's the definition of our molecular definition of a gene? What's our definition of allele? Does it matter if it affects anything in the gene? It could destroy an enhancer site, might create one. Are these different alleles?
This is pretty good age.
All right, get your answer in.
He's so happy.
Let's take a look.
One down, press yes.
Now
That's right, everything.
All right. All right, it's still considered a different allele, different DNA sequence. It is within our definition of a molecular gene, even if it's not in the transcription unit. So it's a different allele.
So I got like, I only answered 3 questions.
All right.
Except for this one.
All right, because our definition of a gene is larger than just the thing that gets translated into a protein, any variation in that stretch of DNA that controls that gene is considered a different allele. Okay? So let's now go back and think about, so in a diploid organism, we have 2 alleles, 2 copies of each gene. How many alleles can there be in a gamete? There's 1 copy of each chromosome, therefore 1 copy of each gene, therefore 1 allele in a gamete. In a diploid, we already covered how many alleles? 2 alleles, 2 copies of each chromosome, 2 copies of each gene, 2 alleles, 2 variants of that gene. What about in a population? How many alleles are there for a particular gene in all humans, for example? A lot. You can have many alleles of one gene. It doesn't have to be just 2. Any individual only carries 2. But in a population, there may be many variants of a particular gene. All right. The last few minutes of class, we're going to translate our formula for looking at gametes from the level of the chromosome to the level of the gene. So if we're thinking about combinations of individual genes, how can those be put together if we assume that they're all assorted independently? How can those be put together into gametes? We're going to play the gamete game. All right, so let me, excuse me. Let me explain a few things so you understand what's represented here. So in genetics, we represent different alleles of a gene by these superscripts. So this is gene AL, for whatever it codes for. Perhaps hair color. All right? Maybe the ability to roll your tongue. Of this gene, each individual is diploid, so they carry 2 alleles. This individual is carrying one allele that is plus, so dominant, and one allele that is minus, so recessive. So how many different gametes can be made if we just consider this single gene and the 2 alleles this individual has? So relative to this gene, how many different gametes?
Is it like you're either going to have one or the other?
Think about the formula we generated for chromosomes. How does that relate to this?
But gametes are like, wait, where?
Think about where these alleles are relative to each other. They're on different chromosomes of a homologous pair.
Different chromosomes. Yeah.
How do those segregate into gametes?
In like a
All right, get your answers in.
But gametes
Is there a question?
because these are homologous.
Everyone have their answers in?
You're either going to have
All right, let's take a look. Okay. The split decision between 2 gametes and 4 gametes. So if you think about 2 alleles, how many pairs of homologous chromosomes are they on? one pair of two chromosomes, but it's a single pair of homologous chromosomes. Right, so one pair of chromosomes in our formula, 2 to the n. How many gametes? So a single gene, 2 different alleles, can only segregate, there's only one way you can divide those up, right? You can end up with a gamete that has the dominant form of that gene or the recessive form of that gene. So the answer is 2. What about if we add a second gene? Another gene with this individual is carrying a dominant form and a recessive form of this gene, so 2 different alleles. How many ways can these 2 genes be put together into gametes? How many pairs of homologous chromosomes these genes would be on? Assume they assort independently. All right, everyone get their answers in. Take a look, and that'll be it for the day. All right, so an individual with these 2 genes having these 2 alleles, they can assort into gametes. And they can make 4 different kinds of gametes. All right, so we'll pick up on Wednesday. We have a few more examples in the gamete game. And they're going to